Hello everyone and welcome to today's presentation titled Revolutionizing Cell and Ex Vivo Gene Therapy Manufacturing, GenScript's Cutting Edge Solutions. This webinar is a part of the 7th annual event in the Bioprocessing Virtual Event Series. I am Melanie Chan of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. I now present today's speaker, Daksha Patel, Senior Segment Marketing Manager of Cell Therapy at GenScript. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Daksha, you may now begin your presentation.
Thank you for the introduction. Welcome everyone to today's webinar. And in this webinar, I will give you an overview of the cell and gene therapy industry. I also discuss the key challenges in the manufacturing. At the same time, I will, through the case study and success story, give you overview how we support the development of cell and gene therapy. And finally, we will wrap up with the question answer session. So let's start with the industry overview. Globally, for clinical use, there are 72 non-genetically modified cell therapy and 33 therapies, which is including genetically modified cell therapy and gene therapy, and 35 RNA therapies have been approved. And additionally, there are more than 4,000 therapies currently in development, ranging from preclinical to the pre-registration. And out of this, like around 2,000 are gene therapy, which include the genetically modified cell therapy, and more than 900 are non-genetically modified cell therapy is in that pipeline. So when it comes to genetically modified cell therapy, CAR T cell therapy remain the most common technology, as we can see here, and that's like and the rest of the therapies are coming in the like TCR, NK, that's coming as the lower percentage. And with over more than 4,000 therapies in development, including many of them is gene and cell therapies, the demand for high quality raw material is growing. As the therapy progresses from discovery to clinical scale, GMP grade material become essential to ensure the compliance and the consistency. However, limited supplier, rising cost, and supply chain disturbance can also delay the production. And top of that, like variability in the raw material can further impact the potency, quality, safety, and that can also introduce extra risk throughout the development. So reliable sourcing strategy is a critical to maintaining the efficiency and assuring the successful commercialization. As we have just seen, the developing of cell and gene therapy like CAR T can be a complex process with challenges like sourcing qualified raw material, managing the supply chain, and meeting all those regulatory requirement can slow down the things, and it can also impact the cost and delay in the clinical application. To avoid this risk, at GenScript we offer integrated solution which tackle this challenge directly, because we providing like end-to-end services from discovery to development work, and that can reduce the risk and make your path to success much smoother. With over 20 years of our global leadership in gene synthesis and biologics, We provide scalable, cost-effective raw material from gene synthesis to the protein, plasmid, antibody, cell engineering, gene editing, mRNA, peptide, and many more. And we maintain the highest quality and regulatory standard, and our strong supply chain ensure the timely delivery of critical material. But we don't just provide the raw material. Our team of scientific experts is here to offer a personalized support also that can help to drive innovation and success at every stage of your project. And GenScript support the increase your R&D efficiency and help you to hit your goal faster. So in this session, I will discuss how GenScript support the journey from idea to IND application, ensuring your cell and gene therapy project move forward smoothly and efficiently. To start, let's look at the standard manufacturing process for CAR T cell therapy. The key step in this cell therapy process include the cell isolation, cell activation, cell engineering or genetic modification, and the cell expansion. To bring this product to the real world market, unique reagent and service are needed, which and depending on the biological manufacturing and the technology use developer, they can select the specific reagent and service they need. For example, the cell engineering step of this process depend on the type of cell therapy. For TIL therapy, immune cell are taken from the patient maybe tumor and put back into the patient, and no genetic modification is needed, so only the isolation, expansion, and analytical reagents are needed. And while if somebody is working with TCR or CAR T cell therapy, the T cells are genetically modified to recognize and attack the tumor. So cell engineering reagents are also needed along with the isolation, activation, and expansion. And first step of the bioprocess is a cell isolation and is commonly performed in wide range of laboratory setting, including for cell therapy and drug discovery research, as well as the core facility as the part of cell manufacturing and cell banking workflow. To support the reliable and efficient solution of isolation process, GenScript offered the cytosine technology is a complete tool which consisting of antigen-specific paramagnetic nanobid and automation equipment. And these reagents are sterilized, non-toxic, available both in RU and GMP grade. They are suitable for use with even fresh or frozen PBMC or leukapheresis and can be used for both depletion or enrichment purpose. The nanobid assuring the compatibility with various downstream applications such as the cell culture, activation, expansion, flow cytoanalysis, or translational research. For automatic cell isolation, GenScript also offers the Cytosine 1000 instrument along with the GMP compatible tubing sets. And this highly efficient and automated equipment is easy to operate and equipped with the key features including user-friendly interface, customizable programming, and data integrity with full traceability. And due to its modular version, it can be fit into the modular workflow and it can be work with any other instrument which required into the modular workflow. So here representative data is presented which demonstrating the performance of cytosine 1000 instrument and bead. In this experiment, CD4 and CD8 cell were co-isolated from the leukapheresis sample using this cytosine technology, and the isolated cell were then analyzed using the flow cytometry analysis. And the data presented here confirm that cell viability is not affected after the cell sorting, and there is no significant difference in T cell purity between the two groups, and the recovery is better with the cytosine instrument, and the vendor instrument has little bit low recovery. So the cell isolation with immunomagnetic bead is a crucial step in the cell therapy manufacturing. but it can be time consuming when it done manually. So streamline this process. GenScript has partnered with the multi-lab to introduce the fully automated robotic-based cell isolation solution. So by combining the multiply lab robotic cluster with the GenScript Cytosint 1000 instrument, it increase the efficiency, but at the same time, it's helped to reduce the human error, and it can also reduce the cost and increase the output. So this platform can be integrated seamlessly with other instruments, so that way one can do manufacturing of the cell therapy in the modular system but in the robotic fashion. uh, So I like to share one of the success story for of who is using this cytosine technology. One of them is a base therapeutic, and the base therapeutic NK cell therapy get FDA approved in the last year December, and that NK cell therapy is used for the treatment of solid tumor. They use the cytosine technology, including the cytosine cell isolation nanobit and cytosine 1000 instrument to ensure the accurate and scalable production of base edited cell. And GenScript cell sorting and activation bead has been registered with the FDA as a drug master file, so that can help to do manufacturing process or IND application. Uh, After the cell separation, the next step coming to the bioprocess is the cell activation. And the GenScript offer the ready-to-use uniform, biocompatible, and non-toxic activation nanobit. In this experiment, enriched T cell were activated with GenScript NC T cell activation reagent, and the one of the vendors reagent keep as a control. And GenScript reagent unable to give high fold expansion while maintaining the stable memory phenotype, ensuring the high cell viability. So in addition, after the activation, there is no need of extra wash to remove the speed. This makes them ideal for scalable clinical manufacturing use. And uh, before discussing the gene editing or cell engineering step, I will first focus on the expansion process and reagent needed for it. Gene editing and cell engineering are key step of the cell therapy, but it's also the complex step. So we will discuss the tools and technique for this is later after the expansion reagents discussion. So recombinant cytokines and growth factor play a very important role in expanding, differentiating, and optimizing functionality of engineered cell in therapeutic like CAR T cell or stem cell therapy. And GenScript offer the comprehensive range, more than 200 high quality recombinant cytokines and growth factor, which is essential for cell and gene therapy. And our products are known for their superior batch to batch consistency, high activity, and low endotoxin level. And additionally, we also provide the GMP platform that allow to um, seamlessly like transition from research grade protein to the GMP grade. And that can ensuring the suitability for preclinical and clinical application. And let's now talk about the cell engineering or gene editing or gene modification. So optimizing the cell function through genetic modification is very important, and it can improve the efficacy, specificity, therapeutic potency, safety, and it can also help to reduce the cytotoxicity. So variety of reagent are needed to achieve this improvement. From discovery to developing stage, researcher using the different tools and different technique. So in this session, I will share how the GenScript supporting the non-viral cell engineering approaches for clinical development and the manufacturing. As we look at this table, um, mm, uh, sorry, As we look at the tools that make this genetic modification possible, one which is stand out among all of them is a CRISPR. The groundbreaking gene editing technology is rapidly gaining very popularity due to the ability to make precise alternation to the DNA. And using this technology, um, cell can be edited both inside the body or outside the body, means ex vivo or in vivo. And traditional CRISPR is already widely used in therapeutic like CAR T for modifying immune cell to fight disease such as the cancer. But the CRISPR hold promise in gene therapy also for to treating or correcting the genetic disorder. And beyond this traditional method, CRISPR is getting more popular in some other area like prime editing, base editing, RNA editing. Even it can use for CRISPR activation and CRISPR interference. So CRISPR is now at the heart of both in vivo and ex vivo therapy, helping scientists to drive innovation and giving the new hope for treating many different diseases. This table shows the reagent needed for different technique. It vary from traditional CRISPRs to prime editing to base editing. Uh, how they work. Due to the time constraint, I'm not going to discuss on detail how each of the technique work, but in this table, I highlighted the key reagent used for each of this technique. And at GenScript, we provide essential reagent to support all these different techniques via CRISPR. So first of them is a nuclease, which are the protein that are play a crucial role in DNA editing and are key material for CRISPR, one of the key material for CRISPR technology. So GenScript provide high quality recombinant nuclease, which is essential for efficient and precise genome editing across the various application. It may be for research or to clinical development. And we have the collaboration with Feng Zheng Lab of the Broad Institute, so we're getting licensing from them also. And our product include different CRISPR-associated Cas proteins such as Cas9, Cas12, Cas13, along with base editor and prime editor protein like ABE, CBE, or PE. And we ensure that the high quality production with the consistent knockout and knock-in efficiency achieved through all of these different nucleus protein. And we offer all these nuclease in both RU and GMP compliant format. So the researcher can use them not only for discovery stage, but they can use them throughout their whole workflow from discovery to development. So the study found that the ESP-Cas9 has a higher specificity than high-fi Cas9 and lower off-target effect compared to high-fi Cas9 product, which is coming from one of the leading supplier, while maintaining the equivalent or superior knockout and knock-in efficiency. And in another experiment also we did the comparison, and the comparison study also reconfirmed that the off-target effect of Cas9, ESP-Cas9, is very low, while the and which has the on-target editing efficiency is similar to the other vendor. So we get licensing this Ultra ESPCase9 from the Dr. Feng Zheng lab, which is a part of the Broad Institute. And in this experiment, human primary T cells were transfected with different kind of Ultra-Cas9. So it's maybe basic GMP, RUO, and GMP grade. And the data indicated that both the GMP, basic GMP, and RUO grade, all the CRISPR-Cas9 maintain the consistent high gene editing efficiency in primary T cell. So RUO products are manufactured under the same process as the GMP grade, and equivalent or better um, performance we can see for all the batch. Like Cas9, Cas12 can be used to create the double standard break in DNA. And here, trying to show that preliminary data with where we use the cell which transfected with the Cas12 enzyme. And in this experiment, different Cas12 variant were used as mentioned like AsCas12a, LbCas12a, MAD7. And the result demonstrate that Cas12a variant shows high knockout efficiency and promising knock-in efficiency, and making it an effective tool for gene editing application in immune cell therapy such as CAR-T or CAR-NK or genetic disorder. So to support this kind of work, GenScript also support like Cas12 circRNA synthesis servings along with the corresponding Cas enzyme. And base editor and prime editor are gaining more attention in gene editing area. However, their complex structure, including multiple protein and RNA component, make them challenging to express and purity. So at GenScript, we successfully produce numerous base editor and prime editor protein, both in high purity and decent yield. So here is one of the experiment which we conducted to evaluate the performance of different prime editing protein and comparing their efficiency and accuracy in gene deletion. So data shows that protein 6D, protein prime editing protein 6C and P6B editing efficiency improve significantly compared to the PE max. And PE7 protein exhibits the highest editing efficiency, approximately 2 times than PE max. And there is no significant difference in cell viability between the group of the different protein used after the electroporation. So to support all this kind of CRISPR-based work, GenScript provide validated Cas9, Cas12, and Cas13 nuclease along with the base editing and prime editing protein, which is ideal for like application of gene and cell therapy and cell line engineering as well as animal model generation and in vitro diagnosis. So you can select the product that best suits for your research or development need from this ready to use Cas protein. And if you are interested in learning more about the base editing and prime editing protein, feel free to connect with us. Due to the time limitation, I'm unable to share detailed data. We have more data also and technical information, but definitely you can reach out us to get the more information. And when transitioning from discovery to clinical or preclinical stage, the need for high quality raw material is crucial. So to meet this demand, GenScript also offer the custom RUO and GMP grade bacterial protein, which is different kind of Cas nucleus, as well as Cas12, Cas13, along with the base editing and prime editing protein for CRISPR application. And all reagents are manufactured under strict GMP guideline and ensuring the aseptic production and traceability. And in addition, GenScript also provide a comprehensive documentation to support the full regulatory compliance. And we so far successfully give more than 25 GMP case protein, and we supported into the more than 9 IND filing, and out of them 5 is already approved. Another key raw material for the CRISPR work is the sgRNA, which direct the Cas9 enzyme to the specific DNA sequence for precise gene editing. And smartly designed Synthetic sgRNA are proven effective for targeted gene editing in knockout and HDR mediated knock-in experiments. So to support this work, GenScript provide chemically synthesized single guide RNA in two different formats. One is a easy edit sgRNA, another one is a safe edit sgRNA. Easy Edit is a more economic solution which has a high editing efficiency and flexible at the scale. While Safe Edit sgRNA is HPLC purified, it can reduce the off targeting and show minimum impact on cell viability. And in addition to this, the free online bioinformatic tools, which is help to designing the single sgRNA and gRNA libraries with our advanced algorithm and user-friendly interface, we providing the free tools to make your own sgRNA design. And that is very in that one, like one can choose the appropriate HDR template or target gene and select the desired sgRNA template, and they can optimize by adding tag for detecting purity or localize the target protein. So its sgRNA design tool is very user friendly and very useful to the customer to make their own sgRNA. And additionally, GenScript also provide add-on QC like macroplasma testing or endotoxin testing or cytotoxin testing if they request. So in this experiment, like sgRNA were designed for three randomly selected targeted gene and synthesis by three different vendor. One of them is GenScript, and their knockout efficiency was evaluated in the cell to compare the performance across the supplier. So comparing the sgRNA knockout efficiency from different supplier, the result shows that the GenScript Easy Edit sgRNA achieve over 80% knockout efficiency for all the three targeted genes, and it's also demonstrated there is a comparable efficiency to other vendors. So the performance of the GenScript sgRNA is quite good. And HDR templates are crucial in CRISPR technology for facilitating uh, um, mm, homolog directed repair during the gene editing. So, by providing the DNA template with the desired sequence, they can guide or repair the process and ensuring the precise genetic modification. So, to make the HDR template, um, it's another key material for CRISPR along with sgRNA. And GenScript HDR template in both linear as well as circular format to support the CRISPR-based application, and that can enhance the accuracy and efficiency of gene editing process. Uh, and each of them has their uniqueness. Like, for example, gene extract is a single standard DNA, which is known as ssDNA also, and is super low in toxicity and making it perfect for T cell therapy. And it can help to insert the length up to 5 kb. Another one is like GeneVend, which is a double standard DNA known as dsDNA, which has the close end and linear DNA, and that help to boost the stability and accuracy. And it's very good for the knock-in application, and it can you with the length up to 10 kb, it's work very well. And for gene circle, dsDNA is a novel but the small circular double standard DNA vector, and it has like minimum 429 base pair backbone, and that's make them ideal for variety of application. And gene circle dsDNA has no antibiotic resistance gene or bacterial origin sequence, so ensuring it cannot replicate itself in bacteria cell means it can't replicate outside its engineered host strain. And this option help the researcher or developer to get efficient gene editing with minimum side effect, and they are designed to scale up for GMP manufacturing. So it can we have the capacity to manufacture them milligram to gram scale. So GeneCircle dsDNA is a circular DNA platform, and that can be also used for use as a transpons vector, and it's offering several advantage over traditional plasmid vector. And here represent the data shows that the gene circle efficiently insert the CD19 CAR uh, GFP knock-in construct around 3.5 kb into T cell using the MF transpon system with the PGK promoter, and we achieved the maximum knock-in efficiency of around 59.89 percentage. So that's approximately 60% in T cell and strong fluorescence expression. And this approach is ideal for delivering the long and complex CAR design. So GenScript commitment to the quality, consistency, and an on-time delivery has made us as a trusted partner among uh, many gene and cell therapy developer and researcher. For example, in December last year, NK, the base therapeutic NK cell therapy get FDA approval, and for the solid tumor. And in this development, GenScript sgRNA they use, and GenScript support them by providing the GMP grade sgRNA for IND filing. So in this development, GenScript not only provide them high quality GMP sgRNA for clinical development, but also offer the comprehensive support in preparing the documentation for IND application. So from concept to cure, GenScript is walk with the customer and helping them to bring their product into the clinical application. And here are a few more examples of successful partnership that highlight the impact of our support in their development. So one of them is Biohang Biotech, which used the GMP grade sgRNA for the first universal CAR T, which approved in China. Another one is the BRL medicine rely on our GMP sgRNA for the first non-viral CAR T IND approval globally. Third one is like T Maximum. Biotech use our GMP sgRNA, and even the Neogen Therapeutic also partner us for the dsDNA for TCR edited T cell. Another one is the Editas Medicine achieve like 90% knock-in efficiency using non-viral DNA template for their sleek gene editing platform. And all this success show how our high quality, scalable solution empower the innovative therapy and driving the success from research to clinical application. So with over 20 of us expertise in nucleic acid synthesis, GenScript has developed optimized process and able to able to implement like strict quality control and extensive expertise in establishing this complete research to GMP level CRISPR nucleic acid reagent product line. So as of May 2024, we have delivered over 120 clinical batches and supporting over 50 plus IND projects received the global IND approval, and many more coming on the way. So we are not just supporting the innovation with reagent and service, we also empowering the clinical manufacturing customer by making uh, collaboration with the instrument company. So to further this goal, GenScript has partnered with Evicta, and Evicta has like Solupor platform, which is automated non-viral delivery system that efficiently deliver various cargo and gene editing tool, including mRNA, CRISPR-Cas9, and plasmid. And this collaboration not only improve the cell therapy by ensuring the high quality modified cell with excellent viability, but this can accelerate the commercialization of non-viral cell manufacturing into fully GMP environment. So we are now expanding our offering with the new service focusing on the PEG RNA. The PEG RNA consists of the spacer, and then it has the scaffold and reverse transcript template and containing the prime binding sites to initiate the reverse transcription. It not only identify the target sites but also encode the new DNA sequence for the edit. And when it's fused with the NIC Cas9 and the reverse transcription, the PEG RNA guide the system to the target site where it introduces a single standard break and allowing this reverse transcription to replace uh, the remove sequence with the new template. So, because of its complex design, PEG RNA is often longer than 110 nucleotide, difficult to synthesize. But however, we have more than 22 years of RNA oligosynthesis expertise, and we offer the desalted and HPLC purified PEG RNA up to 266 nucleotides with over 200 modification options to support your research need. And here showing like our strength in the PEG RNA synthesis area also, like 2 batches of PEG RNA were synthesized with 231 nucleotides. And the MS and HPLC data shows like precise molecular weight for both the sample and has the more than 95% purity achieved for each of them. And this reflect our reproducibility and ability to provide consistent product on any kind of RNA. An experiment shows like dose dependent editing efficiency of PEG RNA. So what's in this experiment, what we did like 141 nucleotide PEG RNA with different dose and different purity. One is dissolved and HPLC was transfected into HEK 293 T cells. And after 3 days, genomic DNA was extracted and analyzed using single sequencing. And editing efficiency reached up to 52% for dissolved PEG RNA and 63.5% with HPLC purified PEG RNA. And this efficiency is increased in a dose-dependent manners, demonstrating that the higher the mRNA dosage, improve the editing efficiency. And another experiment where the editing efficiency compared with different supplier, and in our recent comparison study, it shows that the GenScript PEG RNA with different purification methods show comparable editing efficiency. So PEG RNA synthesis, we doing different nucleotide and they all performing very well in different condition. So in addition to our high quality PEG RNA synthesis service, we also offer the free design tool to streamline your PEG RNA design process. And the key feature of this design tool include like support the knock-in, knockout, and sequence replacement. It's also enhancing the editing efficiency with nicking sgRNA, 3 extension, and silent mutation. And it's quite user-friendly interface, so easily visualize base and amino acid changes with multiple selected options. And if you're looking for validated prime editing system to improve editing or to test the editing efficiency, we have like ready to use prime editing product, which is perfect for quick preliminary work. And we also provide like our PEG RNA synthesis service, which length from 110 to 266 nucleotide with built-in stability modification. And there is no extra cost for that. And we also provide DSALT and HPLC purified sgRNA and complete QC report and that's also within a week. So Cas9 mRNA offered the transient precise CRISPR-based genome editing and minimizing the off-target effect. And to avoid the viral vector risk, this type of gene editing and easy delivery becoming more popular. So we GenScript already started this kind of work, and here I show like comparison of ESP-Cas9 mRNA from different vendor was conducted by evaluating the expression in cell, and GenScript ESP-Cas9 mRNA perform very well compared to the other vendor, and its ready to use IVT mRNA reagents are reliable, high quality, and ensure the research success. And there are certain research shows that electroporation has been widely used for mRNA-based CAR cell generation, but it also causes some high cell death rate and limit the scalability. So to overcome this kind of challenges, scientists also focusing on biomaterial-based mRNA delivery system, including LNP, polymeric nanoparticle, or exosomes, and are as emerging as a superior alternative. To support this shift, we also offer various LNP formulation. Here presented preliminary data for knockout efficiency in different cell line for two different LNP composition. One is LNP LP01 LNP, and another one is ALC031 LNP. And the gene editing efficiency data were analyzed. An LNP formulation with ALC031 LNP shows more than 80% editing efficiency in both Jacquard cell and HEK293 cell line. And the LNP formulation, as we show earlier, like LNP formulation also impacting the editing efficiency. And GenScript, so to support this kind of work, GenScript now offer off-shelf validated ready edit CRISPR LNP solution, which can ensure the efficient and scalable mRNA delivery for your research need. And our comprehensive ready edit LNP solution supports include the Cas9 knock-in knockout, Cas12 knockout, base, and prime editing. So we have like it's more than 11 is, and there are many more is coming, but they can be very good tools for preliminary experiments. And to support the researcher, we also provide the custom ready edit LNP service. And the tailored solution include the enzyme selection, gRNA or DNA template design, LNP formulation, and validation. And that will help the customer to make the research smooth and towards the clinical development uh, progress becoming more easy. So GenScript is always there to help to discover and advance gene editing innovation. And mRNA-based gene editing is growing quickly, and it creating the higher demand for efficient mRNA synthesis and delivery, which is key in cell engineering. And since mRNA synthesis involving multiple steps from gene synthesis to mRNA production to LNP formulation, researchers are looking end-to-end solution which can streamline the entire process. And to address this kind of need and support the customer from A to Z, GenScript offer integrated manufacturing workflow and with high quality and optimized mRNA production. And we also providing like confirm gives like assurance for expression efficiency as well as the QC analysis. And this custom mRNA service package, including like IVT mRNA or circular RNA or self-amplifying RNA or LNP products, and all that's you get from the one vendor, and that's way it's easy to go from discovery to manufacturing stage. And in summary, GenScript's provide comprehensive tools and support throughout your research journey from preclinical to commercialization. And and we also assist the customer for every stage during from preclinical phase to the IND filling and even the BLA phase application. So throughout all your journey, our scientist, our team member, not only providing you reagents, but also providing all the documentation which you need from preclinical to the clinical stage and BLA application. So I'd like to give a quick intro about the GenScript. If you are not familiar with us, we have been around since 2002 and supporting the scientists worldwide. So GenScript is a New Jersey-based life science CRO with global presence, and we offering the wide range of services, instrument, and product to academia, pharmaceutical, and biotech company for drug discovery and development. And we specialize in cell and gene engineering, CRISPR, GMP manufacturing. We helping the CGT developer move from research to the clinical application smoothly. And apart from GenScript Life Science, the GenScript has the another subsidiary company, which is GenScript ProBio, which is a CDMO that's also providing end-to-end service for drug discovery to commercialization in the field of CGT, vaccine, or biologics. And another subsidiary company of GenScript is Lesion Biotech, which is also specialized in cell therapy development, and they get the CAR T cell therapy approved from the US FDA in 2021. And last but not the least, like Best Enzyme is also one of the subsidiary company, which is specialized in synthetic biology product and has established like fully integrated end-to-end system for different kind of enzymatic protein and providing for the agriculture and the consumer food. So GenScript was founded in 2002 and it's globally present, and they have like R&D centers and GMP manufacturing sites around the world, including the New Jersey, Seattle, Singapore, Nanjing, Shanghai. And we are truly global company, and our like more than 200K research papers showing the impact of our product and services on advancing the scientific knowledge worldwide. And our mission is to accelerate the life science research, biologics, and cellular therapy, and ultimately improving the human health. And have we always want to contribute to better improving the human health by providing the different solution. And at the conclusion, thank you for attending my presentation and joining today's webinar. I hope you found it informative and insightful. And if you have any question, please feel free to send your question. Thank you.
Thank you again, Daksha Patel, for your time today and for your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Please visit GenScript's booth in the exhibit hall after the webinar for more information and resources. Questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 2nd, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.